Hello, my dear students. Welcome to a new session of biology. Today we will talk about the cell organelles and we will study all the organelles inside animal cells and plant cells. The key concept in this lesson is eukaryotic cells share many similarities. Yes, as we know, they share many similarities. The main ideas: number one, cells have an internal structure. Number two, several organelles are involved in making and processing proteins. Three other organelles have various functions, and we will study them in detail. Number four, plant cells have cell walls and chloroplasts. Let's start. The cells have an internal structure. Eukaryotic cells are highly organized structures. Yes, these are the eukaryotic cells are the cells that have a nucleus and the membrane-bound organelles, as we studied before. Eukaryotic cells are surrounded by a membrane that receives messages from other cells. These eukaryotic cells contain membrane-bound organelles that perform specific cellular processes, divide certain molecules into their compartments or divide them into smaller portions, and help regulate the timing of the key events like the division of the cell, for example. But the cell is not a random jumble or mixture of suspended organelles and molecules. Actually, certain organelles and molecules are anchored or fixed to specific sites, which vary by the cell type. If the membrane were removed from a cell, the contents would not collapse and ooze out in a big puddle like a small pond. So whenever there is a cell and the cell membrane is removed, the cell will not collapse. Why? Or how does the cell maintains this framework? Let's see. First, we have an organelle called cytoskeleton. Each eukaryotic cell has a cytoskeleton. As you can see, it's like fibers. A cytoskeleton is a network of proteins that is constantly changing to meet the needs of the cell. So again, a cytoskeleton, it's a network of proteins that is constantly changing to meet the needs of the cell. Cytoskeleton is made of small protein subunits that form long threads, as you can see, or fibers that crisscross the entire cell. The cytoskeleton is made of small protein subunits that form long threads or fibers that crisscross through the entire cell. Three main types of fibers make up the cytoskeleton, and they allow it to serve a wide range of functions. The three main types of fibers that make the cytoskeleton, they are microtubules, intermediate filaments, and microfilaments. Let's see. First, microtubules. Microtubules, they are long hollow tubes like the straw. Long hollow tubes that give the cell its shape and act as tracks for the movement of organelles. So this is like the pathway for the movement of the organelles inside the cell. With it, when cells divide, microtubules form fibers that pull half of the DNA into each new cell. When the cells divide, these microtubules, they form fibers, which are called spindle fibers, that pull half of the DNA into each new cell. Second, intermediate filaments. They are smaller than microtubules and they give the cell its threads. Then microfilaments, the smallest of the three. Microfilaments are tiny threads that enable cells to move and divide. The microfilaments play an important role in muscle cells because this is where it helps the muscle to contract and relax. This is a plant cell, as you can see the different organelles. To your right-hand side, this is the organelles found in both cytoskeleton, vesicle, nucleus, nucleolus, endoplasmic reticulum, the rough endoplasmic reticulum, and the ribosomes, and centrosome, endoplasmic reticulum, the smooth part without ribosomes, the cell membrane, the Golgi apparatus, mitochondria for energy, and the vacuum. Then, what you can find only in plant cells: chloroplast, central vacuole, and the cell wall. This is an animal cell. All these organelles that you can find in both, but the difference here is that you can find in animal cells only the centrioles and lysosomes, and we'll study them in detail. Cytoplasm. What about cytoplasm? It's an important contributor to the cell structure. In eukaryotes, the organisms that their body is made of, eukaryotic cells, cytoplasm fills in the space between the nucleus and the cell membrane. In eukaryotes, the cytoplasm fills in the space between the nucleus and the cell membrane. The fluid portion of the cytoplasm, excluding the organelles, so the fluid portion of the cytoplasm without the organelles, is called cytosol, and it consists mainly of water. The makeup of cytoplasm shows that water is necessary for maintaining the cell structure. This is only one of many reasons that why water is an essential component for life. Many chemical reactions occur in the cytoplasm, where water acts as an important solvent, where other molecules dissolve in. Several organelles are involved in making and processing proteins. Much of the cell is devoted for making proteins because this is very important for the cell. Proteins, as we studied before, they are made of 20 types of amino acids that have unique characteristics of size, polarity, and acidity. The amino acids can form very long or very short protein chains that fold into different shapes. The multiple protein chains interact with each other. This almost limitless variety of shapes and interactions make proteins very powerful. The proteins carry out many critical functions, so they need to be made in a correct way. Then we reach the largest and the most important organelle in any eukaryotic cell, which is the nucleus. The nucleus is the storehouse for most of the genetic information or DNA in your cells. DNA is abbreviation for deoxyribonucleic acid. Deoxyribonucleic acid. DNA contains genes that are instructions for making proteins. So the DNA contains the genes that are the instructions, the way how to make proteins. So there are major demands on the nucleus, two major demands on the nucleus. The DNA must be carefully protected, yes, because it contains the information about how to make proteins, so it must be kept protected. Second, the DNA must be available for use at the proper time. Molecules that will damage DNA need to be kept out of the nucleus, yes, for sure, but many proteins are involved in turning the genes on and off. They need to access the DNA at certain times. So how do we keep the molecules that will damage the DNA outside and let the proteins that turn the genes on and off inside or reach the DNA at certain times? The special structure of the nucleus helps meet both demands. Let's see. The nucleus is composed of the cell's DNA enclosed in a double membrane called the nuclear envelope. Each membrane in the nuclear envelope is similar to the membrane surrounding the entire cell. So this is similar to the cell membrane. The nuclear envelope is pierced with holes called pores that allow large molecules to pass between the nucleus and the cytoplasm. Okay, so in this membrane, the nuclear membrane have holes or pores that allow large molecules to pass between the nucleus and the cytoplasm. The nucleus also contains the nucleolus. So this is the whole nucleus. In the central part of the nucleus is called the nucleolus. The nucleolus is a dense region where tiny organelles essential for making proteins are assembled. These tiny organelles, they are called ribosomes. So the ribosomes are assembled or made or created inside the nucleolus of the nucleus. Ribosomes are a combination of proteins and RNA molecules. Then another organelle, the endoplasmic reticulum and ribosomes. We will talk about them together. A large part of the cytoplasm of most eukaryotic cells is filled with endoplasmic reticulum. The endoplasmic reticulum, or abbreviated as ER, is an interconnected network of thin-folded membranes. Interconnected network of thin-folded membranes. The composition of the ER is very similar to that of the cell membrane and the nuclear membranes. Great. Then what? The ER membranes form a maze of enclosed spaces. The interior of this maze is called the lumen. So the inner part between the space, the inner cavity, or the space between the two membranes is called lumen. Numerous processes, including the production of proteins and lipids, occur both on the surface of the ER and inside the lumen. Numerous processes, including the protein production and lipids, takes place on the surface of the ER and inside the lumen. The ER must be large enough to accommodate all these processes. The ER must be large enough to accommodate all these processes. But how does the ER fit inside the cell? You said this is like many foldings. How it fits inside the cell? The ER membrane has many creases or wrinkles and folds. The ER's many folds enable it to fit within the cell, like when you fold your clothes to fit inside your bag. In some regions, the ER is studded with ribosomes, stacked or filled with ribosomes. Ribosomes are tiny organelles that link amino acids together to form proteins. Ribosomes, they are both the site of protein synthesis and an active participant in the process. So they are the place where the protein is made, and they are the active participants in this process. Ribosomes, they are made of proteins and RNA, as we said. After the assembly in the nucleus, we are talking about ribosomes, they pass through the nuclear pores into the cytoplasm, where most protein synthesis takes place. Surfaces of the ER that are covered with ribosomes, they are called rough ER, because they look bumpy when viewed and microscope. As a protein is being made from these ribosomes, it enters the lumen. So the protein is made in the ribosomes, then it enters the lumen. Inside the lumen, the protein may be modified by having sugar chains added to it, which can help the protein fold or give it stability. So inside the lumen of the ER, the protein may be modified by having some sugar chains added to it in order to help the protein fold or give it some stability. Not all ribosomes are bound to the ER. Some are suspended inside the cytoplasm. Proteins made on the ER are either incorporated or combined into the cell membrane or secreted outside the cell. The proteins that are made on the ER, they are either incorporated or combined into the cell membrane or secreted. The proteins that are made on the suspended ribosomes, which are in the cytoplasm, they are typically used in chemical reactions occurring within the cytoplasm. This is how the ER looks like. You can see what is the rough ER and what is the smooth one. Surfaces of the rough of the ER that do not contain ribosomes, they are called the smooth ER, the lower part. Smooth ER makes lipids and performs a variety of other specialized functions, such as breaking down drugs and alcohol. This is very important. Another organelle is the Golgi apparatus. Golgi apparatus, as you can see, they are like many folds. From the ER, proteins move to the Golgi apparatus. Okay, Golgi apparatus consists of closely layered stacks of membrane enclosed spaces. As you can see, they are not like membrane enclosed spaces. That process sort and deliver proteins. So they work on proteins. Its membranes contain enzymes that make additional changes to proteins. Great. The Golgi apparatus packages proteins. Some of the packed proteins are sorted within the Golgi apparatus for later use. Okay. Some are transported to other organelles within the cell. Others are carried to the membrane and secreted outside the cell. So we say that the Golgi apparatus work on proteins. Some of the proteins, the packed proteins, they are stored within the Golgi apparatus to be used later. Some are transported to other organelles within the cell, and the rest of others are carried to the membrane and secreted outside of the cell. Then we move to vesicles. As you can see, vesicles, these are all called vesicles. What about vesicles? Cells need to separate reactants for various chemical reactions until it is time for them to be used. So the reactants, the chemical reactants inside any cell, they need to be separated till the cell need them to be used or need the chemical reaction to be done. In this case, vesicles are here. Vesicles are a general name used to describe small membrane-bound sacs. They are like bags or sacs that divide some materials from the rest of the cytoplasm and transport these materials from place to place within the cell. These are the vesicles. They are like small bags that transport materials or separate materials from the cytoplasm and transport these materials from place to place within a cell. Vesicles are generally short-lived and are formed and recycled as needed. So they have a short lifespan. They are formed and recycled when the cell as the cell needs it. After protein has been made part of the ER, you remember the ribosomes in the ER, part of the ER pinches off to form a vesicle surrounding the protein. The protein can be safely transported from the ER to the Golgi apparatus protected by the vesicle. At the Golgi apparatus, any necessary modifications are made and the protein is packaged inside a new vesicle for storage or transport or secretion, as we discussed in the Golgi apparatus. Then mitochondria. Mitochondria, the singular is mitochondrion. It has an outer membrane and inner membrane with many folds inside, as you can see. Singular mitochondria supply energy to the cell. Mitochondria are bean-shaped and have two membranes. Mitochondria are bean-shaped and they have two membranes. The inner membrane is made of, it has many folds that greatly increase its surface area. So the inner membrane has many folds inside that greatly increase the surface area. Within these inner folds, a series of chemical reactions convert molecules from the body you eat, from the food you eat, into usable energy. Mitochondria have their own ribosomes and DNA, unlike most organs. This fact suggests that the mitochondria were originally free-living prokaryotes because it had its own ribosomes and DNA that were taken in by larger cells. The relationship must have helped both organisms in order to survive. Then we come to the vacuoles. Vacuoles. A vacuole is a fluid-filled sac used for the storage of materials needed by the cell. So this is a fluid-filled sac, a bag filled with fluids that's used for storage of materials needed by the cell. These materials may include water, food molecules, inorganic items, and enzymes. Most animal cells contain many small vacuoles, but in plant cells there is a large central vacuole. This is unique to the plant cell. The vacuole is a single large vacuole. This is the plant cell that usually makes up most of the space inside a plant cell. It is filled with a watery fluid that strengthens the cell and helps to support the entire plant. This is why the plant cells have large vacuoles because it is filled with a watery fluid to support the entire plant to strengthen each cell. When a plant wilt means shrivel or start to die. Its leaves shrivel because there is not enough water in each cell's central vacuole to support the leaves' normal structure. The central vacuole may also contain other substances like toxins that would harm predators, waste products that would harm the cell itself, or pigments that give color to the cells, such as those in petals of the flowers that give them their bright colors. All these can be stored inside vacuoles. Then lysosomes. This is a lysosome. So what's the function of lysosomes? Lysosomes are membrane-bound organelles. Yes, they are covered in or enclosed in a membrane that contains enzymes. Lysosomes defend a cell from invading bacteria and viruses. They also break down the damaged or worn out cell parts, not to keep wastes inside the cell. Lysosomes tend to be numerous in animal cells. Lysosomes' presence in plant cells is still questioned by some scientists, but others assert that plant cells do have lysosomes, though fewer than the lysosomes found in animal cells. So they are more in animal cells than plant cells. Recall that the enzymes are proteins. Do not forget this part. Initially, lysosomal enzymes are made in the rough ER by the ribosomes in an inactive form. They are inactive because if they were active, they would damage the cell immediately. Then vesicles pinch off from the ER membrane, carrying these enzymes, then fuse with the Golgi apparatus. Do you remember the Golgi apparatus? Inside it, the enzymes are activated and packaged as lysosomes that pinch off from the Golgi membrane. Then lysosomes can then engulf and digest. Engulf means swallow or take inside, like this one right here. Look, it takes inside particles and digests the targeted molecules. When a molecule is broken down, the products pass through the lysosomal membrane and into the cytoplasm where they are used again. So this is like recycling. They take in the parts and break them down, and the products pass to the outside of the lysosome to the cytoplasm where the cell can use it back again. Lysosomes provide an example of the importance of membrane-bound structures in eukaryotic cells. Lysosomal enzymes also must be surrounded by a membrane that prevents them from destroying necessary structures, as we said, because the lysosomal enzymes can destroy the cell components itself. This is why they are made in an inactive form. However, cells also uses other methods to protect itself from these destructive enzymes. For example, the enzymes do not work as well in the cytoplasm as they do inside the lysosomes. This is another way to protect the cell itself. Then centrosome and centrioles, another organelle. The centrosome is a small region of the cytoplasm that produces microtubules. It's a region in the cytoplasm that have micro or produce microtubules. In animal cells, the centrosome contains two small structures called centrioles. They are cylinder-shaped organelles that made of short microtubules arranged in a circle. So as you can see, they are microtubules arranged in a circle like a cylinder shape. These are centrioles, a top view, and this is a side view, as you can see. The two centrioles are perpendicular to each other. As you can see, they are perpendicular to each other. Before an animal cell divides, the centrosome, including the centrioles, doubles, and the two new centrosomes move to the opposite ends of the cell. Microtubules grow from each centrosome, forming spindle fibers. Then these spindle fibers attach to the DNA and appear to help divide the DNA between the two cells. We will study this further in the future. Centrioles were once thought to play a critical role in animal cell division. However, experiments have shown that animal cells can divide even if the centrioles are removed, making their role still questionable. In addition, although centrioles are found in some algae, they are not found in plants. Centrioles also organize microtubules to form cilia and flagella. So what are cilia and flagella? Let's see. Cilia look like little hairs, but flagella look like a whip or a tail. Their motion forces liquids past the cell, so they help the cell move. For single cells, this movement results in swimming. For cells anchored in a tissue, this is in a multicellular organism, this motion sweeps liquids across the cell surface. Look here, this is the cilia. They are like small hairs on all sides, but the flagella looks like a whip or a tail for movement. This is the movement, as you can see. These are the cilia to the left, and this is the whip or the tail, the flagella to the right. This is the sperm, by the way. So this is to help it move or advance. Plant cells have cell walls and chloroplasts. This is the last part. Plant cells have two features not shared by animal cells, which are the cell walls and chloroplasts. Cell walls are structures that provide rigid support. The chloroplasts are organelles that help a plant convert the solar energy into chemical energy. Cell walls in plants, algae, fungi, and most bacteria, the cell membrane is surrounded by a strong cell wall, which is a rigid layer that gives protection, support, and shape to the cell. The cell walls of multiple cells can adhere or stick to each other to help support an entire organism like large trees. For instance, much of the wood in a tree trunk consists of dead cells whose cell walls continue to support the entire cell or the entire tree. Even after the cell is dead, the cell walls continue to support an entire tree. Can you imagine that? Cell wall composition varies and is related to the different needs of each type of organism. Let's see. In plants and algae, the cell wall is made of cellulose, which is a polysaccharide, as we discussed in a previous section. Because molecules cannot easily diffuse through or across cellulose, the cell walls of plants and algae have openings or channels to allow the molecules to pass through them. Water and other molecules, small enough to fit through the channels, can freely pass through the cell wall. In fungi, like mushrooms, for example, cell walls are made of chitin, and in bacteria, the cell walls, they are made of peptidoglycan. So this is the difference. What about the chloroplast? Chloroplasts are organelles that carry out photosynthesis. They are found only in plant cells. Okay, photosynthesis is a series of complex chemical reactions that convert solar energy into energy-rich molecules that the cells can use. This is the food made by the plant. This is the sugar. Like mitochondria, chloroplasts are highly compartmentalized. They also have an outer membrane and an inner membrane, like the mitochondria. Chloroplasts also have stacks of disc-shaped sacs within the inner membrane. They are called the thylakoids. They are disc-shaped sacs found in the inner membrane of the chloroplast. Thylakoids contain chlorophyll, which is a light-absorbing molecule, and gives the plant the green color, and it plays a key role in the process of photosynthesis. This is what absorbs the energy from sunlight. These are the thylakoid, the disc-shaped stacks, and inside the inner membrane, and this is the outer membrane of the chloroplast. Like mitochondria, chloroplasts also have their own ribosomes and DNA. The scientists have hypothesized that they were originally free-living prokaryotes that were taken in by larger cells. Both chloroplasts and mitochondria are present in plant cells, where they work together to capture and convert energy. Capture the energy from sunlight by the chloroplasts and convert it into chemical energy. Then this chemical energy is used by the mitochondria to release energy for the cells. The chloroplasts are found in the cells of certain other organisms, such as green algae. And this is the end of our part for today. Hope you understood this part. Thank you and goodbye.